To start our conversation on methods that you might be using in your research at one point or another, we're going to start with a foundational type of sequencing, microbial resequencing, or just known as whole genome sequencing, or WGS. Again, this is fundamental. This is one of the basics of genetic sequencing. It's one of our first goals with the advent of sequencing was to sequence the whole genome of humans. And now we're diving into the microbial world and finding out what is the content of their genomes and what useful things can we find. All right, so understanding this method is a good starting and jumping off point for the rest of NGS and the different methods available. So we're going to focus on bacteria for this service, for this approach, because the case study that we're going to reference is going to be looking into a bacteria, but it's important to note that resequencing or whole genome sequencing can be applicable to all organisms, of course, with the caveat that there is a reference genome. We will be touching on later what you would do without a reference genome. That's known as de novo sequencing. But for now, we'll talk about resequencing, the availability of a reference genome. We can take a look at what is present in our organisms versus what's known. So just a bit of a primer for those of you who are not working with bacteria. So bacteria are single-celled organisms. They're prokaryotes. They're found all over in soil, water, like Charles said, on the table would leave an outline of bacterial microbes. They have a very simple structure, cytoplasm, cell wall, and typically a single chromosome. Although there are exceptions to this rule, and there's exceptions to pretty much every rule with bacteria because of their abundance and ubiquity. So some bacteria have some additional structures, flagella for movement, different villi for attachment. There's also the ability to make biofilms, and capsules can be part of that. They are metabolically versatile, and they're able to survive all over our environment. So because of their ubiquity and because of their abundance and the fact that they're present in so many different environments, we can expect to have some interesting genetic findings in their genomes. So we'll get into that a little bit. Understanding the composition of microbial genomes and the function of their gene products. It's not just important to basic science, it's given us a lot of molecular tools that we work with in other model organisms and still in microbes as well. But it's also important in environmental studies and outside of academia. Microbes are consequential across industry, as we discussed. And it also gives us an insight into human health, where microbes could be actually causing disease, bacteria might be causing disease. And of course, whole genome sequencing, therefore, enables our search for treatments and therapies. Now, I mentioned a case study. We have this paper by Volland et al. These researchers sought to characterize a very unique bacteria. This bacteria grows to about a centimeter long, which most bacteria are 5 to 10 microns, right? So we actually have something that you could almost, you could pick up and look at it, and it's a bacterium. But with such an odd find, they imagined that there might be some peculiarities in the genome. And so how do you go about characterizing an organism like this that is so far from the ordinary? There's a few other things about it that are that are a little bit abnormal. They had membrane-bound organelles that contain not only genetic sequence, but also the metabolic enzymes required for protein synthesis, ribosomes, and other cofactors in there. And the result of the NGS work did eventually show that the unique phenotype of this bacteria and the different, its different behavior, it was reflected in the genome that there were some differences. And so the question here that the reason that we're bringing up this case study is, with something so out of the ordinary, where do you start? What's a good jumping off point? Now, this bacteria is a large sulfur bacteria. We're going to go into the next slide. This bacteria is a large sulfur bacteria, and there are many that have been classified and there are reference genomes available for related species. So now we wanted to compare this new species with what we know. The researchers made the interesting findings and then basically went on to pick a service or pick an approach that would best meet their needs to interrogate the genome without bias towards one type of sequence through amplification or another. They wanted to look at everything. And so microbial resequencing or whole genome sequencing, if you're trying to ask a question like this, what is this species? What is this strain and what is in it? How does it compare to what I know? That would be microbial whole genome sequencing. So what is whole genome sequencing? Whole genome sequencing is a method to determine the complete DNA sequence of an organism. It is, for the most part, unbiased. When you go to sequence the whole genome of an organism, you're looking at everything. You're looking at the coding genic regions, the introns, the exons, the intergenic regions, any repetitive regions that might be in there, and regulatory elements as well. You're ostensibly going to see everything. And so you want to look at how is this different from the genomes that we already have. You're doing a comparison. So it allows for a detailed analysis of the bacteria's genetic information, yes, but not just for bacteria, for other organisms as well. We can look at genetic diversity, evolutionary history, and we can see lineages. We can do lineage studies for population level studies as well. So, like I said, because the ubiquity and the abundance of bacteria and the number of environments that they inhabit, your feed products and their genes could be of interest to us for many areas of study. So if you were to consider taking on a whole genome sequencing project because you have a strain of interest and you want to see how does it compare to what I know, the first hurdle that you would have to get over is successful extraction. The good thing about whole genome sequencing projects is that typically you're dealing with a sample that is more or less pure. You have a culture of something, you've actually grown it in a dish or in a solution. And so this mitigates a lot of the challenges that we're going to go over when we're later when we're talking about shotgun metagenomics. But extraction is an important first step. We've done some research at Novogene, and we found that unqualified samples, we typically find that about 62% of these samples that are unqualified, and when we say unqualified, it means that they're not fit to prepare a library from. It's typically due to amount, and about 62% of samples fall short on the amount side, with about 28%, the remaining 28% is due to some type of DNA degradation from, these can be from multiple sources, but the key is this, because most samples fail because of the starting material, you need to do two things. One, you need to start with enough sample. We have some recommendations for what you can start with, some benchmarks. I won't go over that here, but having enough of your actual solution or your pellet or fungus, enough hyphae, and running that through the correct extraction kit for the material, this is what will give you the best possible chance at conducting your whole genome sequencing experiment. I know, Charles, you have some hands-on experience with this, so I'll just tap you in for a moment if you have anything to add.
Yeah. So as Kyle mentioned, one of the biggest issues is the extraction itself. And one thing we have to keep in mind is where the material is coming from. So if it is a pure sample, we're really just dealing with culture issues. Do I need to remove any sugars or any minerals that may end up in the DNA? Some common things for that would be dialysis kits allow for buffer exchange after extraction. But really, if we're dealing with environmental microbes, our biggest hurdle would be just the uncertainty in the environment.
Thanks, Charles. All right. So to sum this all up, optimize your extraction protocol for your sample type, whether that's a bacterial pellet or plate, a cell pellet, a liquid culture, or a fungal hyphae, whatever you're working with. The best advice is to work with your sequencing provider early on and kind of meet the sample to the extraction kit and meet the efficacy of that extraction kit with the requirements for your library that you're going to be making. And speaking of requirements for your library. So the purpose of this slide is twofold. So one, it's to introduce you to some of the common benchmarks for, well, when you say have enough material to prep a library, how much material does that actually mean? And then also to introduce you to two different approaches to library preparation that we'll talk about in a little bit more detail. Generally speaking, the common whole genome sequencing library requires about 200 nanograms of DNA. That's quite a small amount, actually. But the reason that it's so small is because or so low is because we have to run amplification to get enough material to run that library on the machine, to have enough material to generate clusters on the sequencer. On the flip side, we can also prep a library without PCR, but as I mentioned, we need enough material to run on our lane. And so you need to have the starting material just already there. So you'll need at least 1.2 micrograms to start before the adapters are added. And these are the two different approaches that you can take to library prep. So this will hopefully give you an idea of, if you're considering one of these projects, do I have enough material to even get started? And can I even consider doing the PCR Free Library prep? Or do I need to go the more traditional route of using the PCR Amplified Library? So, sorry guys. So I mentioned the two different library preparations. The image on the slide here is the workflow for preparing a PCR Amplified Library. I'll go through a quick description of what this is. But on Charles's slides in the beginning, we showed the bridge amplification process that occurs on the NovaSeq lane, the flow cell of the Illumina platform. And in order for us to do that process and then perform sequencing by synthesis, we need to have adapter sequences added to the end that are complementary to the oligos that are on the flow cell. So it can actually bind, stay in one place, form a cluster, amplify, form a cluster, and then be sequenced and show as a clean signal on the lane that we can then call as base call, right? In addition to that, this is the step that also adds your indexes. Like Charles mentioned, this is a massively parallel process. You would never take one sample and run it on a full lane of an Illumina NovaSeq platform. It's too much data at one time. For one sample, it would be overkill and just way too much. So you run multiple samples at a time and you index those samples so you can demultiplex bioinformatically later. This process, you're going to start with your gDNA sample, your genomic DNA sample. You're going to fragment it under conditions that favor 300 to 600 base pairs approximately. This is going to yield you two different types of products. You'll get some blunt-ended products. You'll have others that have hanging ends. And so you need to perform end repair so that we have all blunt-ended fragments. Those blunt-ended fragments are phosphorylated at the 5' end, and an A base pair overhang is added to the 3' side. The reason for that A overhang is because that is the mechanism that, well, this library kit uses to add its adapter sequences. You'll see here in the, let's see here, I'll point to it on the slide. So right here in our green fragment, we have a complementary T. I'll point to it on the slide. Right there, it'll overhang the A base pair. We will then introduce ligase and it will be ligated as a complete, at this time, circular construct, right? Circular if it were single strand. From there, we will take the user enzyme and introduce it to the solution, and it will cut at the U base pair, the uridine base pair in the adapter, and thus open up the construct. And what we have here, this green and this brownish sequence, yellowy sequence, these are our sequencing primer sequences. So this is where our sequencing primers will bind so that the polymerase can then do sequencing by synthesis on the actual late once we get to our sequencing step. But that's not all that we need, right? This is not what binds to the flow cell, to the oligos on the flow cell. And so the next step is to introduce primers that contain our P5 and P7 indexes. So on the right-hand side, this is that process. This is the PCR amplification step, right? So through PCR, we ligate or we incorporate these adapter sequences. And then from there, a few extra rounds of amplification so that we have enough material to actually put on the lane and generate clusters, right? So typically, this amplification process, you'll need to do this and make sure that the lane is actually loaded to its theoretical output, because when you load a sequencer, it has to be run with close to the amount of data output it's intended to put out, or else the run could have some things go wrong. So hence the reason for needing a lot of material for library. We're going to contrast this with PCR Free Library preparation. This is essentially the same process. We fragment our DNA under conditions that favor that 300 to 600 base pairs. We do the 5' phosphorylation, the end repair. We add our A overhang, and then here's where it gets different. When we get our adapter sequences added on in this window here, instead of having that circular construct that only has a portion of the adapter sequence where it just has the sequencing primer, now instead we have our read 1, read 2 sequencing primers, the index sequence of our library. That way we can demultiplex later. And it also has the P5, P7 adapter that is complementary to the oligo on the actual lane. And so this is a direct ligation. We directly ligate this on. There's no PCR amplification. And so we make this library that didn't require any PCR amplification, right? Now, why all the fuss about specifying these two things, these two different methods, and specifying why one might be used versus another? The reason comes down to the errors that are inherent with using PCR, or the biases that are inherent in using PCR, and some incorporated artificial error. When you do PCR amplification, there is some degree of bias. Some sequences that are either repeat rich, GC rich, GC poor, they might either amplify preferentially, where you have many, many more of these fragments, or they might not be preferred in the reaction and they'll be underrepresented in your library. So this will lead to a low uniformity of coverage when you go and align these reads to your reference later, right? Sometimes if you do this PCR free library, you'll have some areas with way more coverage than you might have anticipated, and some with way less or none compared to what you were targeting, right? As Charles mentioned earlier, when you're working with microbes, the small genomes, having SNPs that could possibly be introduced by the polymerase during this PCR amplification, it could give you some these artificial SNPs that might be consequential to your research, right? So with having a smaller genome, SNPs are more likely to be significant. And so many researchers working with microbes prefer to use a PCR-free library for that reason. We have a no chance of adding artificial SNPs. And so not only are we having more accurate base calls in this case, and more confident base calls and SNP calls, but we also have high uniformity across the genome. So if you target, say, I want to cover my microbial genome 100x, you can expect with a PCR-free library that most of your regions will be covered at 100x. So as I mentioned, whole genome sequencing and resequencing, it identifies these variants with the use of a reference genome. And so a reference genome is the standard consensus sequence that you use for an organism to call your reads against. Using this method, you can detect genetic differences between your variants. So if you're comparing strains, for example, you can perform transposon fingerprinting. This goes into some methods that might be more applicable in different organisms, but you can do transposon fingerprinting, assess germplasm diversity, and track lineages to in populations. In health and disease, you can map specific traits to specific loci and find out what confers disease resistance or pathogenicity. Through de novo sequencing, you can generate draft, fine, and complete genomes. We'll get into that a little bit later. But right now, these are the goals that if you're going to pursue whole genome sequencing, it accomplishes these goals. And so kind of referring back to our case study here, this is a figure from that case study. You can see when they analyzed the genome and compared it against known genomes of large sulfur bacteria, they found that the assembly was quite a bit bigger than its closest relatives. It's not only was it bigger, but the percentage of coding sequences in the genome was higher, and biosynthetic gene complements too seem to be at a higher rate in this species versus its closest relatives. So getting this kind of data and comparing it across multiple species, this kind of introduces another question. What do I need to compare my sample to others? What is the required amount of data that I need? We talked about sequencing on a lane and having to maybe judge how much data output from that lane that I actually need. But the short answer is it depends on the size of the genome. In this paper, they did a little bit of a survey to see what is the size of the genome, and moving forward, when you go to sequence this, they would target some amount of coverage versus the known genome size to confidently call their SNPs and indels and so on. If you have a reference genome, typically a good target for bacterial samples is about 100x coverage. With a PCR Free Library, you can be pretty confident that this is going to be even. So if you have if you have 1 gigabase of data output for most species of bacteria, this will be enough to cover it at least 100 times. So for fungi, it's a little bit different. They can have pretty large genomes. For fungi, because not only are they larger genomes, but they're a little more complex and can be polyploid, these typically you would cover a little bit higher, so 150 to 200x coverage. So outside of microbes and even outside of this study, whole genome sequencing in general generates very powerful data. Over here, we are depicting a basic bioinformatics workflow that somebody might run their raw FASTQ files from the sequencer through. It all starts with raw data. That raw data is ran through a quality control because garbage in, garbage out. We want to filter things that might be garbage, low quality base calls, adapter contamination where we overread the fragment. And then from there, we map against a reference genome. And using that, we can perform SNP calling, indel detection, SV detection, and CMV detection. Indels are insertions and deletions. SVs are structural variation. CMVs are copy number variations of genes. And this is what some of that readout might look like. When we sequence and look at our SNPs, it's important to confirm the quality of those SNPs. And so whatever bioinformatics pipeline you choose to use, you should get some readout that compares or that looks at the quality of your SNPs in the sample that you actually get off of the sequencer. Validate before you move forward with that finding. As well, you can get some information about the statistics between your samples. How do they compare? What is the size of your mutations and variants in one sample? What is the frequency? And what types of changes are there versus another sample group? And this is all just examples of visualizing that data. Okay. So kind of touching back to our case study here. These researchers did use whole genome sequencing. We kind of talked about the size of the genome versus its closest relatives. They found that it had an atypical complement of cell division and elongation genes, which they believe is at least in part responsible for its dimorphic life cycle, where the centimeter-long bacteria kind of sheds off daughter cells that are a little bit smaller, almost like spores. And so this is a very interesting study and an interesting application of whole genome sequencing because it kind of highlights exactly what it's for. We have an interesting organism that has membrane-bound organelles. It has this odd dimorphic life cycle that's not common, and it's huge, right? There's got to be something interesting there. And so for something like this, where we blur the lines between prokarya and eukarya, it's worth taking a look, and whole genome sequencing provides that objective look into the genome. So very briefly, I want to go over one of the caveats I kind of stated in the beginning of this section. The whole genome sequencing that we've been covering up until this point stipulates the existence of a reference genome, right? Having a reference genome makes everything a lot easier, right? If you have a reference, you can take relatively short reads from the Illumina platform, map it against that reference genome, and have pretty high confidence that you're looking at a gene that is already known, and compare the variation in those reads versus that reference, right? But what if you don't have a genome, right? This is where de novo sequencing comes in. For de novo, it might look a little bit different depending on the species, what you would do to actually build your reference genome. If it's de novo, you pretty much have to build a reference genome to have something usable. And so for bacteria, Illumina sequencing can work. Fungus, it doesn't work as well, that short read sequencing, because it's a more complex genome generally, high heterozygosity rates. And so the need here changes for de novo sequencing. Usually for bacteria, 100x coverage will allow you to identify a vast number of new genes. In whole genome sequencing, doing this, this is also sufficient to map against a reference genome. And just because of the genome sizes of bacteria, it's very easy to get a high amount of data output and coverage. But in whole genome sequencing, where we have a reference genome, we're just identifying fragments of known genes. Whereas in de novo, we're trying to assemble full gene sequences, contigs, scaffolds. So the goal is a little bit different. Because the goal is a little bit different, and because of the limitations and the limitations of short read sequencing and some of the biology of bacteria, or microbes in general, where there are some regions that are highly repetitive, you might require a longer read to assemble a de novo genome. So when talking about de novo sequencing, you might move towards one of those third generation platforms where a longer read would allow you to span regions that are highly repetitive, right? Allow for better ability to assemble long reads into contigs and scaffolds. And also, if you have a sample like a fungus that is known to be highly heterozygous, you'll be able to phase alleles into the different chromosomes too with enough coverage. But for this, you require longer reads. And so outside of this discussion about resequencing, where we have a reference genome, you may want to consider going towards PacBio or long read sequencing, along with possibly some Illumina sequencing to build your de novo reference. The reason that I also recommend pursuing Illumina sequencing for your de novo assembly is because Charles touched on this a little bit. The accuracy of long read sequencing technologies, while it is long read and it's great for assembly, there is some trade-off in the accuracy of the base calling, right? If you're going to phase alleles, you should use something like PacBio Hi-Fi, for example, where there's circular consensus reads that build a consensus sequence that is also a long read. So you have some redundancy there that's built into the long read platform. And then you can validate those different SNPs that you see using Illumina sequencing too, and kind of build your confidence of your base calls by layering Illumina reads.